right. These are the Rugose corals. Shown above are probably not Rugose corals, but we'll get into that at the end. Maybe they are. At least some of them. Yeah. Um, Rugose corals are also called horn corals. They look like that little horn shape that you see at the bottom. What we've got in the big green things are sections through the corals, and there's enough, we think it's probably uranium in there, that it'll fluoresce a little bit as you are taking pictures of them. And so these are composites from dozens to maybe around 100 individual small images, like the way they photograph the moon a little bit at a time, of these wonderful, wonderful Rugose corals. So this is a section crossways, and it's flanked by two sections longways. So Rugose corals. The organism itself is actually fairly compact. It's not very tall, but what you've got is a nice long record of all of the earlier living spaces. So kind of like you get in a mollusk or like a nautilus, some sort of cephalopod. It is going to move up when it needs to grow. And when it does, it is going to essentially build itself a nice new little house up here. So there are things that we're going to look for. What we're going to find are going to be fossils of the coral skeleton. So it's not the animal itself, but it's the skeleton that it secreted. And that's something. There are going to be septa. And so septa are going to be radial, like they go from the edge to the center or center to the edge, but it's actually edge to the center. And they are going to be a structural support. On some kinds of corals, that's all you'll get. You'll have the outer cup, you'll have the septa coming in. On other kinds, you'll have kind of a little mound at the center, and that'll go all the way down through the coral sometimes. On others, you'll have these sort of layers. So every time it grows, you're going to put in a new flat layer for the coral animal to sit on top of. Those are going to be called tabula, like tables. That's what those are. Sometimes there are these other little skeletal elements, and they look sort of like bubbles in a bubble bath, but it's made of the same skeletal stuff as the cup and the tabula. These are going to be called discipiments, running up the side. And we think we understand a bit about the function of each of these things, or at least the evolutionary significance of each of these things. Shown above, this is a coral feeding video, and coral feeding behavior is marvelous to behold. Just absolutely gorgeous. All right, so if we're talking about Rugose corals specifically, what do we look for in their skeleton that will tell us that they are Rugose corals? Well, first of all, you can't really tell one Rugose coral from another if you're looking at the outside of it. Unfortunately, you have to section, and we will section in the class because, yeah, we're all not amateurs. When you section a Rugose coral, you will see the septa inside. And the septa, you're going to be able to orient yourself and figure out, all right, this is where this coral was, this is how it works. So there are going to be 4 main septa. And the neat thing about a coral is down at the pointy end of these horn corals, they're going to preserve, potentially, their earliest skeleton. So it's going to be like one of those organisms where it preserves its earliest parts and then it just keeps growing beyond that. But you can actually look back at that end for a nice life history of at least how the skeleton developed. And the first 4 septa that you will see in a Rugose coral are these 4, and they all have names. So you got your cardinal septa. Think of that as the bottom of the coral. Think of that as closest to the mouth of the coral animal. The mouth's usually going to be in the center on scleractinian modern ones. Then opposite that, opposite the cardinal, you're going to have the counter. And so the cardinal gets labeled C, the counter gets labeled K. Makes perfect sense if you're doing this in German, I think. Yeah. But K counter, whatever, that's easy. And then there are 2 that are coming in from the sides, and those are called alar. Ala means wing in Latin, and so these are the wing septa. And like good wing men or wing women, they are going to be there, and they are going to produce. So when the coral grows, there are only 4 places where it will put in new septa. Because as it grows, it's going to have to make septa, and they can't just appear out of nowhere. So there are septal generation zones. And then they're going to kind of, the other septa are going to be sort of pushed over or slanted a little bit. And so the new septa will fit in. And you can even see this on the side of the corals. It's kind of cool. I'll show you a little later. So you can get new septa at the 4 places where it says new in green. So you can get them on top of the alar septa if you've oriented it with the cardinal at the bottom, and you can get it either side of the cardinal septum. So all of these that are right next to the counter septum, those started out as brand new septa that appeared right next to the alar septum when this thing was still small. So essentially, think of the alars as being like right here, and then this septa appeared out. You can think of the alars as moving down. You can think of the new septa as moving up. Whatever. Nothing moves. They're corals. It's solid. But just, conceptually, if you're trying to get this in your head. I like to think of the new septa as pushing out. But look, they push out and they kind of bunch up here because you can't put any more in because you run into the alar. Yeah. Only the new ones is putting it down here. The other important thing to know about these corals is that septa start small when they're young and they get big when they're old, like everything in biology. Yeah. So when you're looking at a section, you're looking at a moment in time, but if you've got little septa, then those are probably young septa. So spot the little ones and you can orient yourself on the coral. So now we'll go into a whole thing that we're going to do in class. Let me just show you these things. We're not going to really do much with them. We'll maybe do the first 1 or 2. Let's pick out a nice example. Let's do this one right here. So I've oriented all of these so that the counter is up and the cardinal is down. And so in this particular group of corals, the counter septum is massive and it's connected up to this central thing. It's called a pseudo-columella in this particular coral. So the cardinal one's going to be down here, counter is going to be up here, and they're going to be roughly bilaterally symmetric, which means if you can find one alar septum, you count how many septa are in between that and the cardinal, and it should be about that many septa between the cardinal and the alar on the other side. So let's try it. So let's look and see where are our little septa. Like, ooh, look, there's little one, medium, big, enormous. So the little one is a fairly new one. And we know that septa appear either side of the cardinal and on top of the alar. We secretly know our cardinal's down here, so it's not that. And so that means this long one right here, the black thing, that's an alar septum. And there is a brand new septum that's moving up toward the counter side. And so here is your other alar. It's this long one right here. They're usually, the 4 early septa are usually longer or different. Yeah, the alar is often longer. Counter and cardinal will sometimes be smaller, bigger, fatter, whatever. That's going to be a characteristic of a genus of Rugose corals. So that's going to be specific to whoever it is you're looking at. And so here we are. And so now let's count these out. Let's see how we did. So here is cardinal. And if we go 1, 2, 3, 4, 5, number 6 is the alar. So, okay, if this is cardinal, we'll go 1, 2, 3, 4, 5, 6, 7. So not bad. And look, there's one brand new septum coming in here beside the cardinal. And that's the one that's thrown into soft. So that's why there's 7 on this side. We put in an extra one down there. Not too bad. You will also see sometimes that the septa around the cardinal septum, it's quite often a little shorter and a little fatter, and the septa around it will sometimes make like a little hoop like that, where they're each reaching in and trying to touch. It's like they're all bowing down to the cardinal septum. Yeah, that's kind of cool. So practice these on your own. We will also practice them in class. You will also section a coral and you'll get a whole lot of practice that way. Let me tell you. Here are the same ones. They're oriented, like higgledy-piggledy. Have a go. See what you can do. try it out on these. Now, these are all oriented correctly or consistently. So the cardinal septum should be down and the counter septum should be up. What can you tell? Can you spot the alar septa in here? And can you tell me what's unusual about the cardinal septum? What's unusual, if anything, about the counter septum, etc.? These are all meant to be the same species. And when we've got a line of them, these are the same coral, and so we're cutting it at different places. So we're looking at different points in the life of this individual. And so if you can't see which septa are new in this image, you can go back to the previous one. You can count septa, and then that may tell you, that may give you a clue. Because the alars aren't really going to move, and the counter and cardinal aren't really going to move. It's all the other things that fill in around them as the cup widens. Here's a different kind. Can you spot the septa? I think this one is actually, pretty easy to diagnose. And it makes a lot of sense. This one is actually doing a good job of bilateral symmetry if you get it oriented right. So if you're at home looking at this on an iPad or something, that's great because you can turn it around and say, here, obviously, this is the orientation. If you're downloading the PowerPoints, it's just a little thing on the screen. I think you can probably rotate it around and make yourself happy. Different kind of coral. There are often going to be thickened septa, and quite often it's specific to the cardinal side. So it'll run from cardinal to alar on one side. They'll all be thickened, and then they'll all be kind of skinny on the other side. That's a particular group of corals that do that, but they're a group that's pretty common in the Pennsylvanians, so I happen to have a few of them. Yeah. Here are some other kinds of corals. These are all corals that the authors believe they have oriented correctly, and I would tend to agree. And so they'll put little triangles at the cardinals at the bottom, counters at the top, and the alars are on the side. So this is like a coral with the answer key already on it. So looking at this thing, are those the right positions? Can you support those based on what you see? Remember, they're looking at different slices from the same coral, so you may not be able to see a new bud of septum in a particular slice, but if you look at the previous slice, usually the previous letter, then you may definitely be able to see that. So I think these are all the same one, and I think these guys are all slices from the same coral. So that can help you if you're losing count. Yeah. All right, so that will be a fun activity. And we will pause there, and then we'll go for Rugose corals part 2, where we get into all the biology of these things, at least as far as we can tell. I'll leave the same mascot up there, so see you in a minute.